We have with us Dr. Takeshi Egawa as our first speaker of our joint event from Washington University in St. Louis. Dr. Egawa's lab has been working and contributing significantly for many years now in deciphering and understanding the development and function of lymphocytes, mostly CD4 and CD8 T cells and B cells, and their hierarchy, their clonal expansion, both in the viral infection settings and also cancer settings. The lab has been focusing for many years on how we can have a long-lasting immune response, and also focusing, which is also part of today's talk, in different subsets of lymphocytes and how we can salvage their different features instead of taking a whole heterogeneous population of T cells or B cells. So as you know, the way that we're basing the talks with Open Box Science is that we're inviting a first or last author of a recent paper that has been very impactful. And we're talking on this and the research beyond. So today we're going to be talking about CD4 cells. And Dr. Egawa, thank you so much for being with us. I leave it up to you.
Okay, thank you very much. Thank you, Anna and Christina, for a generous introduction. And I really feel honored to be just contributor for the student-driven organization. This is a good thing. It's a nice product of COVID time, so maybe we should just make the best use of this platform maybe in the next coming years. So today I'm going to talk about some recent papers. I try to explain a background beyond what's written in the paper. So that's maybe I'm going to try to do. Then let me share screen. This one. Do you see the slides okay?
Yes, I think we're now fine. Yeah.
Okay. Let me put the pointer here. All right. So today I'm going to talk about how maybe immune response can be maintained for sometimes weeks, sometimes for a month to fight against some persistent antigens. I think most of people study this kind of biology targeting CD8 T cells, but we try to do something different from other people. So that's why we also try to go into the CD4 T cell side, as well as we are also doing some other things in B cells. So the first thing, I have no competing financial conflict of interest for this talk. Okay. So we've been interested in these immune cells, particularly called the lymphocytes, because they are fascinating cells in terms of the power of the proliferation. So typically, basically, you are almost ready to fight against any infections, maybe including COVID, but depending on the frequency of cell to start with, typically you have a very small number of T and B cells which can recognize particular pathogens. Then upon, say, challenged by infection, so you have to expand these cells, which exist in like 100 to 1,000 in the steady cells to tens of million cells within a week, so that you can contain infection. Particularly, this is very obvious for the number of CD4 and CD8 T cells that expand during the infection, which is relevant to real important immunity, triggering both cellular and humoral immunity. Then maybe this is, you have seen this chart. So this is the maybe kind of a T cell proliferation kinetics upon so-called normal infections, or the flu infection, or we in the mice system, we say CMV infection, or similar to immunization. So when virus infects, so maybe the cells start to compete against viral expansion, then you need to simply override the replication of virus to contain them, then clear them. Eventually, this happens within a week. Then eventually you have good memory for some years. So this is a very important thing, and also recognize, re-recognize during the COVID time. Then during this time, so they can divide maybe up to probably 5 to 6 times per day at the peak of response. So basically, they can divide every 4 to 6 hours, almost for a few days, to achieve this high clonal frequency. At the same time, the cell acquire so-called effector functions, including cytokine production and the maybe killing functions in some cases. which is a very important functions, but it's typically inevitably causes basically shortening their life. So basically they spend energy, they do the professional functions, but typically these cells go away relatively quickly, which is also important to contain basically the contract inflammation so that you don't have to have a unnecessary tissue damage. So this is typically fine, but in some infections, you have to, you have to deal with much longer duration of infections. In human infections, I think maybe most famous chronic viral infection is probably associated with CMV or EB virus or hepatitis viruses. Then in mouse world, so maybe Christina should be very familiar with, but Rafi and many other people use this virus called LCMV to study this, both in acute and chronic infections. But there's one strain of the variants of LCMV, which is called the clone 13, which acquire 2-point mutations compared to the original Armstrong strains. So this replicates very fast. So this changes the game between the viral replication and the immune cells. So in a clone 13 infection, which replicates so fast, they establish very high viremia in the normal situations, which cannot be contained by expanding CD4 T cells, CD8 T cells, or even the antibody responses. In this case, what happens is basically cells are exposed to like a consistent, continued activation for a while. Then also some altered inflammatory conditions. So in this case, cells are not able to handle the virus quickly, but they basically change the strategy to keep the basically immune response for a longer duration at a relatively low level. Then you can maybe appreciate by the number of CD8 T cells shown here. The white dots is shown for the normal infection or acute infection. So the virus doesn't replicate so fast, but T cell can replicate fast. Then at this peak point, so maybe most of the virus will be eliminated. The immune response is going back to the steady state and stay as a memory cells. But in this chronic infection, so what happens is the virus goes up pretty high within a week or two. Then what happens is the T cell, instead of keep expanding, because that causes some tissue damage, maybe the immune system take some sort of alternative approach to attenuate some immune response, but continue these recognition of antigens and also maybe some effector function, so that eventually you can slowly and maybe combine all the arms of immune responses, including CD4 and CD8 T cells, as well as antibody response to eventually control the virus. Then in this particular infection system, so in the mice can control and eventually clear the virus, spending 3 weeks or 3 months instead of like a 10 days to 2 weeks. But this is a very important system, but we don't know much about the real mechanism by which this continued low-level immune response is sustained over a month, over weeks sometimes. And also, this phenomenon is relatively close to what we can see in the tumor microenvironment, in which tumor cell persists in case of humans for years, but the human immune system try to contain or try to prevent the growth of tumors so that the maybe tumor are not going out of control. Then eventually, this system is important in many different ways, and this led to the discovery of anti-PD-1 checkpoint blockade therapy as well. But we want to know basically how this continued immune response can be maintained through multiple cell types. Then, so the question that we ask in this particular paper, or just I try to talk about today, is how the sustainable immune responses, particularly in the T cell side, are maintained in the face of persistent antigens, probably through different mechanisms compared to the acute infection context. And more specifically, what are the similarity and dissimilarity between CD4 and CD8 T cells? Because CD8 T cell response for this context have been studied in many different groups, and there are numerous papers, but there are relatively fewer papers which studied about the CD4 T cell response for weeks or long term. And how are they different from canonical immunological memory, which is induced by a normal vaccine or the acute infections? Then also maybe one question that we can ask is, people have studied a lot about the CD8 T cell exhaustion for many years, but they are not much known about the CD4 T cell behaviors in this context. So maybe a simple question we can ask is, do CD4 T cell get exhausted in these similar conditions? Then to do this, so we combine two different ways. One is the more like a dry biology or dry, biomedical analysis, using the single-cell omics approach to define dynamics of CD4 T cell responses to this chronic viral infection using LCMV, as well as some sort in a tumor microenvironment. But this is an important thing. So it's a maybe power of modern biology, but we also want to always appreciate what's really happening in the biological validations on top of this, data analysis time point. So we use some biological experiments to validate the finding in a single-cell omics approach using the conditional knockout approaches. So we've been working on this quite closely with a group at Stanford led by Anshu Sapathy, who actually did the MD PhD at the WashU, and actually he worked in my lab for a little bit. So we basically set up this collaboration in the past few years, working on both CD4 and CD8 cell immune responses against the infections and also in the tumors. Then we extract as much information as possible from this dry analysis. Then we validated everything or most of the stuff in vivo, mostly in the lab, mostly driven by two graduate students used here. She just graduated and she's back into med school. Then Saran Raju, he's now doing a fellowship in infectious disease environment. So for the sake of time, I'm not talking too much about CD8 T cells, but maybe you can go back to many reviews. But this is one review taken from Nature Review Immunology published in 2019 by a consortium PI describing what's known about these persistent T cell responses against tumors and infections. And we people discover this very interesting population of progenitor exhausted T cells, which are the cells actually responding to anti-PD-1 checkpoint blockade in the context of infection tumors. Then we also have done some work on the CD8, maybe defining a kind of a different hierarchy of precursor cells, which are the same cells starting with the TCF-1 positive progenitor exhausted cells. But we also found some sort of intermediate precursor cells, which are important to maintain. And also we try to look at how the population dynamics happens in the context of this exhausting conditions using single cell analysis as well as the biological analysis. Then this is a recent paper. We did the more like a heavy the single cell analysis to find out what the epigenetic and the gene expression profiles that you can see in the face of chronic infections. If you're interested, please refer to these papers. I think I'm not talking about this one today. But today I'm going to talk about CD4 T cells. Compared to CD8 T cells, which are simply becoming naive to activated killer cells, the CD4 T cells are a little bit more complicated. Then maybe as you know from many lectures or textbook or many publications, so CD4 T cells have maybe two major distinct functions. One is function as effector cells producing cytokines, then acting on many innate cells or some other adaptive T cells, such as Th1, Th2 polarization happening in the context of some cytokine milieu. But also another important function of CD4 T cells is giving a help to B cells in the germinal centers, which drive affinity maturation of the B cells, causing a somatic copy mutation in the germinal centers, eventually establishing sterilizing immunity mediated by B cells, and also super long-term immunity through plasma cell differentiation. So these are two major functions of CD4 T cells, which is more complicated compared to the CD8 T cell, more like a simplified differentiation to naive cells to effector cells. But what's interesting is that maybe these two functions are not necessarily segregating in the middle. Maybe this is very early segregation phenomenon, which is maybe reported by Casey Weber's group a few years ago, that maybe CD4 T cells, depending on affinity or depending on the signal, such as IL-2 receptor signaling, so CD4 T cells basically bifurcate their terminal fate into either this TFH path or the maybe effector or TH1 path in case of viral infection, even before the first division. So, which is totally different from CD8. And also, this is also validated by single cell analysis, showing that there are reasonably distinct non-overlapping clonality between the CD4 becoming T effector cells or the T follicular helper cells. So that maybe this bifurcation is really happening in very early events. So this added maybe another layer of complexity to think about how CD4 T cell can be maintained for weeks. Then, for example, one way to think about this is maybe there may be some sort of independent precursor cells, which can maybe specialize to TFH compared to the T effector cells, maybe there may be a different pool of cells, maybe which can maintain these processes. Or maybe we can even take a step back, like a CD8 T cells, we think that probably effector cells, differential effector cells are short-lived. So they need to be replenished by this so-called precursor cells or like a memory-like cells. But in case of CD4, maybe these cells may be even long-lived. They may not even have to be replenished all the time. So this is a totally open question at that point. Then also maybe another kind of the model that we can think about is maybe something similar to CD8 T cell happens. Maybe there may be some sort of precursor cells which can be maybe bipotential to give rise to both T effector cells and TFH cells in the middle of the infections, which maybe is quite distinct from the very early event, which initially generate this effector and the T follicular helper cell directly from naive with the independent clonal populations. So, and also maybe another question is, are they really respond to anti-PD-1 like maybe CD8 T cells? Maybe this kind of a question was totally unaddressed a few years ago. So we decided to take this part using the power of single cell analysis as well as the some biological validation experiments. So then our analysis is very straightforward. So we simply use this well-established viral infection system, SMB acute infection and SMB clonality infections, which establishes two different patterns of antiviral responses in the C57 black 6 mice. Then simply take the antigen-specific T cells, CD4 T cells out of this animal with a couple of different time points and dump everything into the single cells to see what we can see. And this is maybe kind of a summarize the entire concatenalized data showing what kind of clusters, what kind of heterogeneity we can see in the single cell analysis. And this is pulled everything from the acute and chronic and a couple of different time points. So you see all the cell types that we will see in this analysis. Here, one maybe obvious end terminal fate of CD4 T cells is the Th1 effector cells. So this is shown in the green light colors in this plots. The other end is the TFH. So this is helping the B cells. But what we can see is that there's clearly a lot of cells in the middle, either going this way or the other way. But we simply use the representative gene expression to define or guess which cluster corresponding to the known populations, like maybe Th1, TFH is T effector cells maybe enriched for the expression of CXCL6 and interferon gamma, as well as the transcription factor BRIMP1, so which is kind of hallmark feature of T effector differentiation path. And compared to that, the TFH enriched for another chemical receptor called CXCL5 and BCL6, which is an important factor for both B and T cells in the germinal centers. Then there are some cells in the middle which are marked by TCF1 or TCF7 gene, which encode the TCF1 proteins. So it's complicated, but also some other markers like a SLAM-F6. So these are maybe getting to some sort of big populations. Then this is maybe corresponding to so-called steady state memory cells. But there are clearly some other populations which we do normally see in acute infection context. Then we initially look at the frequency. So what are the distribution we can see in a different way between the acute infection, chronic infection, maybe relatively early time point or relatively late time points. In the acute context, so we can see a nice Th1 differentiation shown from these cells, as well as the TFH response, which is shown here. Then over time, after the virus is cleared by like 3 weeks after infection, at this point, there's no virus remaining in this infection context, the vast majority of cells acquire lasting memory phenotype, which is the dark orange phenotype, which is shown here. But compared to this acute, some sort of ideal response by CD4 and CD8 to the viral infection, in a chronic infection, the picture looks slightly different. So first, maybe early time point, there are clearly more cells which are kind of showing close to naive, but they still get activated, kind of weakly activated cells happening here. And there are much fewer cells becoming this Th1 effector phenotype compared to these conditions. Then virus persists long time after this. Eventually, at the steady state time point and 3 weeks after infection, we can see this memory cells and the maybe smaller fraction of Th1 cells compared to this time point, but bigger fraction of TFH. And also there's another populations which are slightly different from acute infection memory. It's more like an activation, mixture phenotype of activation and the resting memory phenotype, which I'm going through in the next slide in more details. So this is, again, the day 21 comparison. So in the acute, so it's a good immune response, ideal immune response compared to this some sort of altered immune response against chronic infection here. So you can see a couple of different findings. So one is there's a clearly a bigger fraction of cells acquire this light orange color, which is a mixture of activation and the memory phenotype. But also, even in this orange population, which appear to be the same based on this clustering, but we can see a quite distinct gene expression patterns. So this is the basically the volcano plot showing a differential expressed gene from acute memory cells here compared to chronic memory cells, only showing this resting memory phenotype cells. Even though this is thought as a resting phenotype, so we can see many genes which are associated with activation happening. Somehow this is the cell sitting, but some kind of mixture of activation phenotype, although they still maintain a memory phenotype, which is similar to what people have found in a CD8, the stem-like or CD8 progenitor population. So we thought that maybe this may be some interesting population to look at, and also there's clear differences. Then this is maybe some sort of summary what we can see as a comparison between the chronic and acute. So we see a different pattern of expression gene differentiation of CD4, and predominantly driven the cells into memory phenotype, regardless of antigen clearance. But also we see this activation phenotype, activation memory phenotype, which is relatively specific to clone 13 infection, which is with the antigen persistence conditions. But even within the resting memory, we see a quite different gene expression patterns happening in these conditions. Then, okay, single cell is great, but we want to validate in a more sophisticated level. So we want to test many knockouts. So we can't run single cell for everything. So we try to deduct this information to identify some sort of markers or protein expression that we can apply in the flow so that we can do a much deeper analysis, or sometimes we can get the live cells. Then maybe after several attempts, so eventually ending up with this two transcription factor expression is useful to define these populations. For example, this is the expression of the BCL6 protein and the TCF-1 protein. So these two basically protein expression can segregate the population into mainly three populations at the two different time points from the clone 13 infections. Then what we have seen, and also based on, which is also consistent with the literature, is that there's a cells which BCL6 high population, which is maybe considered germinal center TFH cells, which helping the germinal center B cells to acquire affinity maturation happening. And this is the TH1 cells which lose TCF1. Then vast majority of cells acquire some sort of memory phenotype, which contains both activated and the resting memory phenotype. And we further confirmed this by based on the expression of, for example, memory specific gene, which is maybe CCL7 or the SLAMF6. So these are mostly enriched and more highly expressed in this memory type of populations. Then CXL5 and PD1 is the highest in the TFH. Then CXL6 and CD150, which is also called the SLAM, and the BRIMP1 reporter gene, so which is also highly enriched for the TH1 compared to the other populations. So maybe our guess is true, and also this is consistent with the single cell analysis. Then there are some literature, and also they're suggesting that maybe TFH can be a memory cell, so there may be an overlap, or maybe another way, people looking at BCL6 expression, sometimes only looking at TFH, sometimes only looking at the memory T cells, but not so many papers compare two different populations together, which are also marked by BCL6 expression. The one thing we try to do initially is, what if the TFH can be the real memory cells or precursor cells to maintain the responses of long-term CD4 T cells? The one thing we simply did is try to look at the mice which are lacking B cells. So this is a mutation of immunoglobulin heavy gene. So these mice don't have any B cells. In this case, first, we can see a continuous CD4 T cell response. So B cell is not necessary. Also in this mice, this population is missing. So this is also consistent with our guess that this is a germinal center TFH cells, which are totally absent in this animal. But we can see this memory population here. This means that probably these populations are distinct, maybe at least in this LCMV infection conditions. Then the power of single cell analysis is maybe you can predict the differentiation path based on the similarities that are large scale PCA analysis, or maybe which is called a pseudotype projection analysis. So then we can connect the individual cell events dot to guess what are the development pathways which connect the cells into different pathways. So in this process, we only recluster the cells from a 3-week time point, only from the chronic. So this is the one terminal fate effector Th1 cells, this other faded TFH. So based on this computational predictions, what we can see is there's an overlapping path up until this point from the early memory was close to naive cells getting here, then this is the bifurcation point, either going to the Th1 and TFH. So probably there should be some common precursor cells sitting somewhere in the middle, which can give rise to both EFH and Th1, rather than there's a distinct population of cells happening here. This is also confirmed by gene expression patterns here. Then we also want to see what are the real epigenetic changes happening, or the epigenetic are different, and also what are the transcription factors involved in this differentiation process. Then we combine this with the ATAC sequencing using the same data analysis. Then in the same way, we initially guess what are the corresponding population, or what this correlation between the RNA sequencing data and ATAC sequencing data. Then eventually maybe concluded that number 7, this purple population, is similar to resting memory. Then yellow population is maybe more like a transitional or the mixture of activation and memory phenotype here. Then on the right side here, so this 4 and 5 shown here is the TFH clusters, which is marked by high CXCR5 expression. Then 2 and 3 are marked by high T-beta expression, which is more like a Th1 effector cell type here. Then we did the same way. So we do some further analysis or projection analysis. So then it's a trajectory analysis starting from number 7 population to either TFH or Th1. Then we can see something similar here. So initial transition is common between the two pathways from 7 to 6 happens, which is associated with the activation of many genes associated with the transcription factor binding sites of AP1 complex, NFAT, and the NF-kappa B pathway, which is obviously downstream T cell receptor binding by antigens. So initial transition from 7 to 6 appear to be common between the two pathways, which is driven by antigen recognition, or release of antigen recognition, release of suppression by PD-1 is possible too. Then after the cell get into 6, so some cells going to turn to the left to become TFH, which is associated with acquisition of mainly the basic helix-helix transcription factor, which is maybe associated with many differentiation processes, including TFH differentiation, driven by transcription factor ACL1 and 2. So this transition happened. Then on the other side, some cell turn to the right from this 6 to 2 and 3, which is more associated with the RUNX and TBX transcription factor binding. Means this TBX family contains TBET, which is the master regulator for Th1 differentiation. And also RUNX is also known to cooperate with TBET to drive the Th1 differentiation. So again, it appears that there should be some sort of common different developmental hub giving rise to both TFH and T effector differentiation in this context. Then, so these are biological, it's maybe kind of a in silico analysis, and we can get many more information out of this. But eventually, as a like an immunologist or the classical biologist, so we want to test what's the biological meaning of these findings. Then, so what we try to do is, okay, we know BCL6 and TCF marks these populations, but unfortunately, we can't sort the cells with intracellular sense. So we try to find out the surrogate marker for these two expressions. One is maybe Li108 or SLAMF6 as a surrogate for TCF1 molecule, and CXCR5 as a surrogate for BCL6. Then we were able to segregate these 3 populations by combination of these 2 markers. Then what we did is simply infect the B6 mice, then collect these cells based on these markers 3 weeks after infection, the time point we use for most of the analysis. Then adaptive transfer these cells into infection match animal, which are also infected with the same virus at the same time, with a different congenic marker, so that we can keep track of the fate of transfer cells, and which can be compared to the endogenous response which developed in this host animal. So this is a busy slide. So, but just I want to pick up only a few major time points. So our definition of these progenitor cells is that they should be able to expand much more than terminal differentiated cells. So the most easy or obvious readout is that how much expansion we can see from individual each of this donor population. Then this is maybe shown as the representative FACS plot here, and also statistics showing here. Then this memory-like cells that we define, so this is the same as the TCF-1 positive BCL6 negative cells. So these cells expanded by far the most compared to either differentiated TFH or differentiated T effector cells, meaning that these have a highest proliferative cells in the upon transfer context. And also, this is the endogenous response, so we should see the same across the 3 different group of mice. These expanded cells also give rise to the differentiated TFH cells and TH1 cells, almost a similar frequency compared to the endogenous cells. This means that maybe they have they retain the high proliferative capacity as well as the multipotent, or at least the bipotent precursor potential to give rise to 2 different functional subsets of CD4 T cells. So then, also then we went back to this single cell data because this analysis is all population based. So we need to transfer like 100,000 cells to the donor cells to make sure we can see the events. Then even if we can see 2 populations from the same donor pool here, it doesn't mean that individual cell contain the bipotential, the progenitor potential to give us both TFH and TS. This could be maybe interpreted as a mixture of cells. There may be some distinct population which only give rise to TH1 or which only give rise to TFH. So then we went back to the single cell analysis and looking at T-cell clonality, which is a kind of endogenous fate mapping tool so that we can keep track of clone, which originally exists in a low frequency, then expand to eventually give rise to these terminal fate. So then basically we reanalyze the single cell data to assemble BDJ and CDL3 for T-cell receptor alpha and the beta chain. Then we identify a few hundred clones. Then eventually what we can see here is compared to this previous knowledge about this acute infection, in which we can see a quite distinct T-cell clonality between this TFH arm and T effector arm, or TH1 arm. We can see most of the clones are found in multiple populations, and most of them contains both on this arm and this arm. And the most obvious way is, so we simply pick up among this a few hundred clones, so we pick up maybe top 10 expanded clones based on the frequency. The majority of these clones, as you can see here, are spread into all these 4 populations of cells, including the terminal T effector and the TFH fate, as well as this resting memory and active memory cells, which means that probably instead of these 2 fates are maintained by independent, separate precursor pool, but it's more reasonably to speculate that there should be some sort of common precursor cells which give rise to both TFH and T effector cells after maybe a certain time point in these chronic infections. It's also confirmed that maybe these precursor cells that we see in the single cell as well as the biological experiment have a bipotential cells which can give rise to TFH and T effector cells. Then, okay, so these are all good experiments, and also transfer experiments are very powerful too, but sometimes you can see an artifact because these cells should not be taken out of the body and injected into IV. Then we try to determine whether these cells are really necessary to maintain the response. Now, even though sometimes we can see like a transfer experiment which shows that transfer cells can do blah, 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 but not necessarily these cells are important. So we try to look for a way to specifically deplete this population to see if what we can see in this context. But then we try to first develop a key driver conditional knockout mice in which we can deplete the gene only in the CD4 T cells, not touching CD8 and some other T cells. Because this is a viral infection context. If you touch CD8 T cells, they're mostly affecting the kinetics of the virus that we try to avoid to minimize the artifact. Then among several genes that we tested, so we ended up with the CD4 T ligand as a driver for Cre, then which now cross this Cre mice to BCL6 conditional knockouts so that we can deplete the cells, deplete the BCL6 gene only in CD4 T cells. Then what happened in this animal is that, so we can see almost normal number of antigen-specific T cells after a week after infections. Maybe there may be some trend of a small reduction here, which is consistent with the loss of like a memory precursor cells and the TFH cells, which are known before. So, but bottom line is, these mice can give rise to Th1 population, which is negative for TCF1, negative for BCL6, almost at a normal magnitude compared to the wild type animal, which is we use the Cree mice alone without the BCL6 mutation here. So this is good. But what happened in the next is week after infection, week after this time point, so we now see almost total loss of these Th1 positive cells. Of course, these mice don't have these memory cells or TFH cells. So this means that there's a loss of Th1 populations. So again, so the point is, we have 3 populations in normal conditions. Then one assumption is that maybe this population should give rise to Th1 after a certain time point. So this data indicates that initial wave of Th1 is coming independent of BCL6, which is consistent with the previous finding, and which is maybe consistent with many other findings. But after a certain time point, maybe after day 10 or 2 weeks after infection, now this Th1 cells has to come from this memory-like populations, or there may be possibly a TFH, some sort of TFH intermediate cells, which indicates that at least this population, BCL6 positive cell transition to BCL6 negative cells, is happening in these conditions. And this is a necessary process to maintain the entire CD4 T cell responses against this persistent antigen. Then the next we did is, okay, how long this can be functioning? So how long maybe this stem-like population or precursor population or memory-like population, we can just name them in many different ways. Are they really long-lasting cells to support CD4-dependent memories? So initially we thought that CD4 depletion, then transfer the cells would be a good way, but this also causes a lot of problem because the antibody is hanging around for a long time. So we utilize this BCL6 conditional knockout mice, in which we can see initial wave of CD4, but this cell disappear 2 weeks or 3 weeks after infection. Then what we did is simply infect this animal, then wait for 3 weeks. At this point, there's almost no endogenous response happening here. Then we can transfer the stem-like population from congenic animal, then wait for like 2 weeks, 3 weeks, sometimes 4 weeks to see how long they can support the CD4-dependent immune responses. Then we've used a couple different readouts, but maybe easiest readout is looking at the germinal center responses, which is totally dependent on T cells, the CD4 T cells. Then, so we compare again the same way, transfer so 3 different populations, effector, progenitor cells, and T effector follicular helper cells, transfer into this BCL6 conditional knockout mice. Then what we can see is only the mice which receive these memory-like cells can maintain germinal center responses like 3 or 4 weeks after this transfer of cells. At this point, there's no endogenous cells remaining, so this is only supported by these transfer cells, which is also statistically shown here. So this maybe also indicated that maybe these cells at least can last for at least a few weeks to continue to support the entire CD4 T cell responses, including B cell health. Then finally, a lot of people ask how this is similar to the tumor response. Of course, the infection is the most important thing, and we know that the antiviral immunity is so important through the experience through the COVID time. So we also have to deal with this infectious pandemic maybe coming every 10 years, every 20 years. So that's an important finding. But also the other way is, many people suffer from cancers. Then people think that this chronic response against T cell response against chronic antigens is also important for tumors. So we try to look at this tumor-bearing anti-tumor response of CD4 to see if we can see any similarity or there may be some dissimilarity between antiviral and anti-tumor responses. So this way, in this case, we did some sort of similar experiments. In this case, we used the basically transplantation of overexpressing tumor cells in combination with the OT2 CD4 T cells, which recognize this overpeptide expressed by the tumor cells. So then we're also looking at the different tissues because the LCMV infection is a systemic infection. So we can basically look at any tissues, including blood or the spleen. Maybe we can see a very similar picture across different tissues. But in case of tumors, typical tumors are localized. So we can see a tumor, but also most immune response is triggered by the draining lymph node. So we also look at the tumor draining lymph node. Then using the same marker here. So what we can see is the CD4 T cells can be found in roughly 3 populations, maybe mainly in this memory-like phenotype cells in the tumor draining lymph node. But in the tumor environments, most of the cells are already terminally differentiated to the effector cells. Because this tumor doesn't form a tertiary lymph node structure, so we don't see TFH populations in this. This is specifically this tumor model. Then we try to see if there's anything relevant to this chronic viral infection models or that relevant to a tumor response by CD8 T cells. Then one experiment we did is simply CFSC label this OT1, OT2 T cells transfer right before the tumor implantation to see how they behave in the tumor context. Then, so this CFSC dilution here, then one thing we can clearly see is the terminally differentiated cells in the tumors are fully divided. Maybe they diluted the CFSC almost completely normally here. And but in the precursor cells in the tumor, in the draining lymph node, also show a similar degree of proliferation. So this is not necessarily remaining naive phenotype. So they recognize antigens, they divide, but they acquire some sort of memory phenotype here, and which is also analyzed by combination of CFSC and some sort of markers, including TCF-1, BCL6, and the CXL6 as a marker for this T effector differentiation, and also activation marker PD1 and CD44. Then in many papers looking at CD8 T cell response to tumors, there are a lot of controversy whether this stem-like CD8 T cells are residing in a tumor or they must be coming from the lymph node. And there are maybe, could be depending on the tumor or depending on the tumor tissue themselves, depending on the patient. I think there's no clear conclusion about this. But at least in this model, what we can see is the tumor tissue do not have this memory precursor phenotype. All the cells which we can find in the tumor environment is all terminally differentiated. Then maybe one way we can try to dissect in deeper on this analysis is looking at the differentiation in a draining lymph node. So for example, this is the more careful analysis of TCF-1 and the CXL6 expression. This is the marker for TH1 differentiation. Then based on this analysis, what we can say is the vast majority of CD4 T cells, which recognize tumor and undergo division in a draining lymph node, retain this memory-like phenotype, but small fraction of them initiate this T effector differentiation before losing TCF-1 or before migrating out of the tumor draining lymph node. Then eventually only the cells which finally terminally differentiate or acquire T effector differentiation phenotype are migrated to the tumor tissues and maybe sitting there to help maybe CD8 T cells or maybe helping DC or making cytokines to control tumors, which may be slightly different from the CD8 T cell response. But I know this is a kind of premature analysis. We can do a much better, much deeper analysis on this one in the coming studies. Then, okay, so sorry, I may be rushing a little bit, but try to summarize the take-home message here. So these CD4 T cells are a little bit more complicated than CD8 T cell responses because they need to have generate 2 different terminal effector phenotype, including TH1 type and TFH type. Then initially this bifurcation happened independently during the priming by DC at the very early time point. So which is also supported by distinct non-overlapping clonality between the 2 fractions of the CD4 T cells. However, once the antigen persists for a longer time, then immune response is a little bit different from the normal optimal responses. This response is maintained by the common precursor cells. And actually these differentiated T effector cells and probably TFH as well. So these are short-lived. So you need to supply, we need to basically replenish these populations which are dying out from the precursor population, which are common in the bipotential precursor cells, which are recruited by T cell receptor stimulation. And supporting this evidence, we see now no overlapping T cell clonality to pretty much almost complete overlapping clonality between the 2 arms of CD4 T cell responses over time. And eventually this precursor population is showing some similarity to a CD8 progenitor CD8 T cell populations, but probably slightly different behavior in terms of tissue distribution, or also triggering to differentiate into pathways. Then maybe we may see something similar in a tumor microenvironment, although I think we still need much more sophisticated experiments to conclude for this analysis. Then so finally, I want to thank people who contributed the work. This is mainly done by former MSTP student Yu Shir. She's back in med school, also initiated by Sharon Regis, another MSTP student. And finally, completed by Sunny Shin, who's a postdoc in the lab. And also a close collaboration with the ancestor Patty's group at Stanford, who had a quite a good expert in a single cell omics analysis. And also we got many help from our community in the WashU, including Ken Murphy's lab, and also the Bachelor lab and the Corona lab, and also this the funding. I want to thank funding from NIH and the Leukaemia Lymphoma Society and Cytokine Cancer Center. And I want to thank your attention. Then if you have questions, I'm happy to answer all questions. Thank you.
Thank you so much. That was amazing and a lot of work for sure. I'm sure people have worked several hours in the lab. So yes, everyone, if you have questions, you can write them on the chat and I can read them out loud. We already have a question from Natalie Edner. Do you think these precursor cells also play a role in propagating autoimmune responses?
It's a good question. Probably yes, probably no. But probably we have to think is this LCMV infection is a very unique system, very useful system, very established, but it causes some sort of systemic inflammation. So in this case, there's almost no naive cell remaining in the infected animal. But in the autoimmunity, probably yes, or there could be some thymic output happening during the autoimmunity. That's also happening. But bottom line is, I believe so, but we haven't done any experiments. Maybe similar answer may apply to antimicrobiota response in the intestine as well. Also, in this case, the T cells are exposed to bacteria which are sitting pretty much steadily in the microbiome, in the lamina propria, or in the face of epithelial layer in the gut. So they are exposed to some sort of same stimulation over time, maybe similar to autoantigen happening in the autoimmune context. Then probably the answer is yes, but we don't have evidence. We haven't gone into the autoimmunity model. I think this is clearly one of the future directions that we want to pursue.
Yes, I also see Geraldine has a similar question. Very elegant work. Congratulations. In addition to infection or tumor models, do you see progenitors in autoimmunity? And there's a second question, I believe. Does the differentiation and maintenance of progenitor-like T cells require certain cytokines?
We don't have definitive answer, but probably similar to memory. I think maybe IL-7 and 15 should be. We look at I'm not sure we have looked at the IL-15 knockout or the IL-3 receptor knockout conditions, but again, so this could be clearly one of the future directions that we want to pursue.
Thank you, Dean. I will jump in myself for a couple of questions too.
Sure.
Yes. You also mentioned about cancer being, of course, every cancer setting is different, so we don't know exactly how this population will be. Have you compared different models at all? Do you have any idea if a more slow progressing or fast progressing tumor model can have differences in this population, or a subset of a subset maybe?
Yeah. It's a good question. Then there's a huge limitation in the tumor model. So there are not so many tumors that we can, for which we know the CD4 epitope.
Yes.
So that's the biggest limitation. So we had to use this overexpressing tumor model transplantation. Then the biggest issue is this tumor basically rejected 2 weeks or 3 weeks after transplantation. So which is a kind of big limitation. So maybe we wish we can find the endogenous CD4 epitope in some tumors. Then if the tumor persists for like maybe more than 3 weeks, or it could be a month, and that would be really interesting conditions. Then we try to develop some system, but I don't think we have a clear answer for this. I'm sorry for that one.
Yeah, for sure. Yes, and as you mentioned for the tertiary lymphoid structures, I think if we could have like a model that is inducing TLSs, because B cells are also important, as you also said, when we're talking about CD4 cells. we might be able to see a little bit more there. I'm gonna finish with a what's next question.
Yeah.
Maybe therapeutically wise, in a similar way to the CD8s, do you think that these progenitors will be more responsive? How we can salvage their features?
For this one, so one maybe obvious question is how we can induce them. It's same for the anti-tumor immunity, or how you can target them in case of autoimmunity. So that's maybe just the beginning. So we don't know, and also we need to test maybe a few different models to think about this. So that's maybe one question. But the other question I'm personally more interested in is why you need this type of structure. So when T cell expand, the B cell expand, so eventually they terminally differentiated, they mostly die. That's an important process for you because you want to keep old cells around. You need to try to keep new cells. But probably that could be a limitation in some context. So simple questions. Every single cell type divide and going through divisions, they eventually cutting their life off. That's a kind of bad thing for like a stem cell biology, but that's a very important for tumor suppression. But our goal is simply we want to dissect out the pathway, gene expression pathway, what limits their life, then maybe override this in the context of CAR T or something. In this case, we can keep stimulating T cells, but they don't lose their life. Then if process is longer, maybe useful to apply to like anti-tumor therapy, both in CD4 and CD8 T cells. That's maybe one of the important things. And the same for your generation. So young cells should basically kick out old people to do a good science. Same for cells. Old people should, like me, should disappear at some point, but you guys can take over the entire immunology world, the science world.
Thank you. I think we have no more questions in the chat. Thank you everyone for being with us. Thank you, Dr. Egawa, that was amazing. And for anyone who missed it, or also have the recording of it, and we're now upload it on the OBS YouTube channel. So thank you everyone, have a wonderful day, night, and see you in the next one.
Enjoy summer. Summer is
Yes.
coming.
Thank
Hi,
you.
Jerry.